Good evening and welcome to Season 5 of Lightning Talks. We are coming to you live from the Seattle Aquarium on the shores of Puget Sound in the traditional and contemporary territories of the Coast Salish people. And tonight we have speakers joining us from across the country, from California to South Carolina. And wherever you may be joining us from tonight, I'm so excited you're here with us and hope you enjoy the show. My name is Nora Nickum. I use she, her pronouns, and I'm the Vice President of Conservation Programs and Partnerships here at the Seattle Aquarium. And tonight, our theme for this episode is Ocean Ancients. Now, you met some in the trivia for those who were engaging in that, and I wanted to introduce you to one more really cool ocean ancient before we get started. This is a coelacanth, and scientists used to think that coelacanths had been extinct for 65 million years. They had found fossils and that was it. And then in 1938, they found a live one. So it turns out these ocean ancients are still around. There are two species in the ocean today, one near the Comoros Islands and one in Indonesian waters. And fortunately, they're protected from trade. So hopefully they will stick around for many, many more years to come. Now, if you are new to Lightning Talks, let me tell you how it works. We have 5 speakers, and they each have 5 minutes, not 1 second more, to share one thing that they're super excited about. And they are going to get warnings as they go along, so they know if they're getting close to time. So they'll get a warning at 1 minute remaining, another one at 30 seconds left, and then one when they are at time. And if they're still going at that point, they will get this. And that is why it's called Lightning Talks. So, of course, we always kind of hope that at least one speaker will get us a lightning bolt. Now, we hope that you will participate in the chat or use this QR code or link to submit questions as we go. I'm sure all the speakers will get your curiosity raised. And a bunch of you have already warmed up in the chat with the trivia question. So after everyone has presented, we're bringing all our speakers back to the Lightning Talk stage for a fun discussion. And as part of that conversation, we're going to bring in a bunch of your questions. So please send those in as you think of them. Now, if you enjoy this free science event tonight, we hope you'll consider a donation to the Seattle Aquarium so that these events can remain accessible to all and we can make great advances in our mission to inspire conservation of our marine environment. So without further ado, I'm going to go ahead and introduce our first speaker, and we'll get this party started. So Dana Stoff joins us tonight with a solid argument for why cephalopods are the new dinosaur, and I am very ready to be persuaded and can't wait to hear why. So Dana is a science communicator and a marine biologist with a PhD from Stanford University, where she studied baby squid. How much cooler could it get? Dana also is the author of some fantastic books for both adults and kids, including one called Monarchs of the Sea, The Extraordinary 500 Million Year History of Cephalopods. And that certainly explains why she is here with us tonight on the Ocean Ancients episode. So Dana, are you ready to get started and talk to us about cephalopods?
I'm so ready. Thank you for having me.
Your slides look great. Go for it.
All right. So cephalopods are the new dinosaurs, is my argument here. And believe it or not, you're already primed to agree with me because the promo image for the Ocean Ancients show is a fossil of cephalopods. And Geordie LaForge agrees with me. Dinosaurs have been overplayed. It's time for cephalopods, which is the group of animals that includes octopuses, squid, cuttlefish, and a lot of their extinct relatives, to step onto the stage. But I am going to start by talking about dinosaurs because dinosaurs are a triumph of science communication. And I'll even manage to advance my slides. So think about it. These are not living animals that you can find in your backyard or on a hike. The only reason we know about dinosaurs at all is science. And that science has been communicated so well that dinosaurs feature in our most beloved media. Dinosaurs are really, really cool. But everything that's cool about dinosaurs is also cool about cephalopods. Let's start with how incredibly old they are. So this is a diagram of the geologic history of Earth. We have the modern times on top with the truck. And as you go down, we're getting older and older. Those numbers on the side are how many millions of years into the past we're going as we travel down. And so here on this slide, we're seeing the oldest dinosaur, Nyasasaurus, which showed up in the Triassic about 230 million years ago. It's a long time ago, but the earliest cephalopod, Plectronasseris from the Cambrian, showed up about 490 million years ago. So cephalopods are twice as old as dinosaurs, and I would say deserve twice the respect. Let's take a moment to really appreciate Plectronasseris. Plectronasseris is this really cute, weird little cephalopod. It was actually small enough to fit in the palm of your hand and had a shell that looked kind of like a wizard's hat. And now cephalopods are mollusks. They're related to snails and clams and other things with shells. And all of these early cephalopods had shells too. And the shape of their shells reminded scientists of goat horns or other horns, which is why a lot of cephalopods have the word ceres in their name because it means horn. And that's actually similar to dinosaurs as well. A lot of dinosaurs have saurus in their name, which means lizard. And just as a little reminder what we're here for, we're all kind of tired of the sauruses, and we're here to catch up with the cereses. Okay, you're thinking Plectronasseris, it's cute, it's kind of cool, it's kind of funky, but it's small, and you're like, Dana, dinosaurs were really, really big. That's why they're so cool. Fear not, because I understand how big dinosaurs were, and I understand how cool that is. And I'm here to tell you that while they may have been some of the most gigantic animals ever on the planet, they were not the first gigantic animals on the planet. The first giant animals on the planet were, in fact, cephalopods. And when they evolved, there was nothing big. It was all little scuttling trilobites and worms and things. And then the early cephalopods did this. This is the fossil of a cephalopod called Endoceras that is thought to have grown to maybe 20 feet or maybe even longer in length, the first really giant animal on Earth. And scientists love these fossils. They love to argue about how they might have swam in the water, whether it was horizontal or vertical. There's some evidence going both ways. Maybe they even rotated a bit between them. And I love this argument because I think that both options look completely ridiculous. And that brings me to the next thing that I think we all know and love about dinosaurs, which is that they're pretty goofy. This is what makes them so much fun, right? They have those plates, they have spines, they have thagomizers, they have duckbills, they have helmets. Well, cephalopods got pretty silly too. They grew those shells. The classic ammonite shell is a coil, of course, but they grew them in all kinds of shapes. And in fact, this is one of my very favorites that I'm about to show you. It's called Diplomaceras, and Diplomaceras grew its shell in the shape of a paperclip, hundreds of millions of years before Clippy was invented. Diplomaceras got really big. These are like 5-foot-long paper clips. And as if that weren't weird enough, they also lived for a very long time. Scientists have estimated possibly an individual might live as long as 200 years in the ocean, swimming around, doing its giant paperclip thing. I really wish these still lived in the ocean today. Which brings us to one of the last very, very cool thing about dinosaurs that everybody loves to learn, and that is that they went catastrophically extinct when a giant meteor smashed into the planet. And cephalopods did too. Now, I know you're saying we still have cephalopods today. Aren't there octopuses in the ocean? And it's true. We also still have dinosaurs today. That's all of the chickens and pigeons and all of the other birds that we know and love. So really what happened is that these animals went extinct.
Thank you, Dana, for giving us the lightning bolt.
So I'm so glad I could oblige.
Please finish your sentence. What really happened was they went extinct and
they left us with a few descendants who squeaked through and are blowing our minds with their amazingness today.
Amazing. Well, I love this idea of the sauruses are so yesterday, and now we're going to talk about the cereses, and they're amazing. I'm wondering, the Endoceras, the one that was 20 feet long, how do you think the ocean would be different if we still had that amazing being with us roaming the seas?
So they just look so cool. And they are thought to possibly have been scavengers instead of predators. We don't even know if they had jaws. So they might be just and they lived in relatively shallow water. So can you imagine, like, in in water shallow enough to snorkel, maybe you might just see one of these absolutely gigantic animals just kind of slowly oozing along with its huge, because they couldn't move fast with those huge shells, and it would just have a bunch of arms out, kind of like, like picking up detritus and anything else it could find.
Amazing. I love imagining that. Well, I hope everyone will put some questions in the chat for you. I just have so many, so I'm sure our audience does too. So thank you very much, Dana. And I'm going to introduce our next speaker on the Lightning Talk stage, Rachel Kalisperis. So Rachel has been with the South Carolina Aquarium for 25 years, and she has a Master's of Science in Marine Biology. And she splits her time between designing really fun, interactive exhibits that optimize animal care and doing field conservation research. And tonight she's going to share some of that field conservation research with us on an animal, a species that is so ancient, it is often called a living fossil. So Rachel, we're getting your slides up and they look great. Take it away.
Okay, thank you so much. Thanks for having me. I will be talking about the Atlantic horseshoe crab, Limulus polyphemus, and our work with the South Carolina Department of Natural Resources to help monitor their population here in South Carolina. This is such an engaging species to me. I love it. They're in our touch tank here, as you see, at the South Carolina Aquarium. And they're actually fairly common in touch tanks and aquariums because they're relatively harmless. They have a hard shell or carapace that protects them. Their feeding claws are relatively weak pinchers. And their tail or telson is not used for defense. It's actually used to flip themselves over if they get turned upside down. And they're part of the Ocean Ancient series today because they've remained relatively unchanged in the fossil record for millions of years. They're of incredible importance for so many reasons. They're food for endangered species such as loggerhead sea turtles. Their eggs are a critical food source for several migratory shorebirds such as red knot. And their feeding habits, they dig through the sediment with those legs. They stir up the sediment, which is an important ecosystem function, similar to you reworking your garden soil at home. They support a commercial fishery because they're harvested for bait used in the eel and conch fisheries. And they're really important for human health and safety. Their blood, which is blue, is used to create a test that checks medical equipment and IV fluids or IV medications for bacterial contamination before they enter our bodies, making sure that we're safe. So with all of these incredible, important demands on one species, it's important that they be monitored constantly and carefully and managed well. And to do that, researchers need data, data such as how many crabs are there and where do they go? Where do they like to nest? And that's where we can come in and help gather some of those data for the researchers to use to protect the crab and ensure our fisheries are sustainable. So let's talk about the nesting piece for a bit. This is a nesting pair. Now, typically the crabs spend most of their time in deeper water, but females will come on shore to nest in the spring at the very highest tides. So she'll come on shore, she'll lay her eggs. The male, which is attached to her back, will deposit his sperm on top of the eggs as she walks out of the nest. Often you'll see more than one male trying to fertilize the same nest, and those are called satellite males. They come on shore in mass numbers to nest. And we were fortunate enough to capture this video during the day this past year. Typically, the crabs prefer to spawn and come up and nest at night. They prefer the nighttime high tides. But we were lucky enough to capture this, tag a lot of crabs, and help document this location as an important spawning habitat. The eggs themselves are very small. They're about the size of quinoa. One female will lay 80,000 to 100,000 eggs in one season. She'll come up to nest a few times. They have a slight greenish color. They'll incubate in the sand for 2 to 4 weeks. And after that, they'll hatch from the egg, emerge through the sand granules, and swim into the water column where they'll stay swimming for about 21 days. Then after that, they'll settle down to the bottom where they'll spend the rest of their lives. And they're just like adults in miniature form, but without the tail. So next, I'd like to play a 1-minute video for you that our marketing team created that really sums up and shows you the work that we did in the field, helping some researchers gather these data for the horseshoe crab. Like I said, this is at night, so you'll see us working with headlamps. It's really fun work and important work. Here we're getting measurements. Measure them at their widest width. And that's the tag that we'll put on them that helps researchers know where they go. This is an acoustic tag. During the day, we look for the eggs. Another way to find out where the crabs are nesting. Like I said, they're small. That's a 1 millimeter sieve we use to find them, and those are those hatchlings emerging from the sand. All right, and with that, I'm done.
All right, Rachel, no lightning strike for you. Thank you for sharing those amazing videos. You know, I've never met a horseshoe crab in person, but now I really want to. I kind of think that they just look like hard hats. So I was surprised by that initial video showing how flexible they are with their tail. And if I remember correctly, there are some in the touch tanks at the South Carolina Aquarium that people can come meet.
Yeah, for sure. That was the video from our touch tank. So you sure can.
Awesome. Yep. Can you share maybe a highlight or two from your most recent research season with us? What are some of the things you've been learning?
Well, this past season in particular, we were able to explore a few different habitats looking for the eggs, and we were able to document them at a site that they hadn't been documented before. So, you know, because they do come up at night, you can miss them. You know, it is dark, and by looking for the eggs during the day, we were able to find a location where they hadn't been observed nesting before, and now we know another spot that they're nesting. They because of things like habitat loss, they have to look for new nesting areas. So it's important for us to keep track of where they're nesting.
Wonderful. Well, thank you so much. And a reminder to everyone to put your questions for Rachel in the chat. And Rachel, we'll see you back here again in a little bit to answer more questions. So now we're going to move from a living fossil to an actual fossil. Our next speaker is Cindy Loy. Cindy is a plant ecologist at the University of California, Berkeley, who has been known to say she has tumbled down the rabbit hole of deep time. And she has a lot of different interests, but a common theme across them is looking at how plants and plants communities respond to environmental change. And tonight she's going to share a story about an ancient, really cool fossil found right here in Washington State. So Cindy, take it away.
Okay. Thanks. So these iconic northeast kelps that you're seeing here on the screen are keystone species for dense marine underwater forests. Let's see. Yeah, and these forests form the base of complex, highly stratified ecosystems, and they provide refuge and food for hundreds of invertebrates, fishes, marine mammals, and seabirds. And one of the more famous inhabitants are the sea otters, who wrap themselves in the kelp blades before falling asleep. So after storms, we often find stranded kelps on the beach. And if you dig through the pile of blades, like I'm doing here on this picture, you might be able to find the stipe that the blades are connected to, and at the base of the stipe, a holdfast. So the purpose of the holdfast that you can see in this inset, as the name suggests, is to anchor the kelp to rocky substrates. So the kelp holdfasts themselves harbor a lot of small organisms as well. So they form a kind of miniature ecosystem on their own. So this figure shows the relationship within the brown algae plotted against time. This diagram which is based on DNA analysis, shows that the ancestors of modern kelps, the kelps are indicated by the red bar, evolved during the late Cretaceous. That's about, roughly 100 million years ago. Fossil proof is, however, very rare. There are 3 small red dots in the diagram, and they mark the only groups that we have fossils for. And one of those red dots is a kelp. So one of those red dots that I just mentioned represents this roughly 13 to 14 million year old Jules Crania fossil that my collaborator Rosemary is holding. It was the oldest kelp until now, and only one of 2 specimens. And it's pretty big, right? And with an air bladder and basis of the blades, it looks actually a lot like modern kelps, like, for instance, elk kelp that you can see on the picture on the left. So knowing how rare these fossils are, you can understand why my collaborators Jim and Stefan here were very excited when Jim found a fossil holdfast on the Olympic Peninsula. So Stefan is holding one of them. And you can see darker sheet-like structures with nubbins near the margins of the rock. And these fossils are 32 million years old, so more than twice the age of Jules Crania. So why do they think these fossils were made of kelps? Well, almost all kelps have holdfasts with finger-like structures. They can range from long ones like Laminaria on the top right to ones with nubbins like Postelsia in the top left. And these fossil holdfasts, here we have one in cross sections, were attached to a variety of shelly organisms. And in this case, a large oyster that you can see more to the bottom of the image. And the stipe of the fossil was not preserved. So when you trace the darker outlines of the structure, you can see sheet-like structure coming off from the central part. And more laterally, they form the finger-like structures we saw earlier. So what do these fossils tell us? Well, let's have a look at what we know about the organisms that appeared with them. So on the bottom of this diagram, we see a timeline going from 40 million years ago on the left to the present on the right. And above that, we see reconstruction ocean water temperatures. And we can also see that our holdfast on the left more than double the fossil record of the kelps. Black and green lines under icons of animals show the ranges of associated organisms. And we think our early kelps were likely relatively small and might have been grazed upon by hippo-like marine mammals called Demostillions. There's no fossil evidence that the early kelp ecosystems were very rich. We know for sure that the canopy-forming kelps developed at the latest during a period called the Miocene, when we see Jules Crania appear for the first time. That is also when we start to see the gradual appearance of more modern kelp forest inhabitants, starting. Haha, did
All
it.
right, Cindy, high five. Love the lightning. I think you were talking about how the ancient kelp was different from today's kelp. So maybe you can just tell us a little bit more about that. Were the sizes different, and do we know why?
Well, there are three reasons why we think they were small. So you saw the icons of those Demostillions, and they're reconstructed just based on their bones as hippo-like animals that likely were hovering slowly at not so deep water, walking on the seabed, and then if you do that, that would be ideal for grazing relatively small kelps. Another reason is because none of the fossils we have the stipe attached, and that probably indicates that the stipes were relatively flexible and soft. And to form dense forest with upright stems, you need a stiff stipe. So because the stipes are missing, we think they were actually not that sturdy, and the kelps were not that big. So yeah, that's why.
So interesting. Well, we certainly love our kelp here at the Seattle Aquarium, and it's fascinating to learn more about how long kelp has been around, even if it doesn't look exactly the same as what we research and enjoy today. Thank you very much, Cindy, and I'm sure there'll be some questions in the chat for you, so we will look forward to welcoming you back soon to hear some more about fossil kelp. And in the meantime, I'm going to introduce our next speaker, Kramer Wimberly. Kramer is a master scuba diver trainer and a lead instructor with Diving with a Purpose, which is a community-focused nonprofit dedicated to the conservation and protection of submerged heritage resources, particularly African slave trade shipwrecks. He is also the creator of a coral ecosystem monitoring and ocean restorative healing program at Diving with a Purpose called CARES, Collective Approach to Restoring Our Ecosystem. And Kramer, we are very excited to hear what you have to share with us today. We'll get your slides up there, and they look great. Take it away, Kramer.
Okay. Good evening. All right. As a scuba diver for 40 years, I've seen significant changes in the marine environment. Right? I designed this programmatic way to address the challenges that I've witnessed over that 40 years. Diving with a Purpose Collective Approach to Restoring Our Ecosystems, or DWP CARES, is a citizen science-based initiative that teaches divers and non-divers the importance of a delicately balanced ecosystem and the negative impact that human beings are having on that marine habitat. DWP CARES is a coral ecosystem monitoring program that collects data on key indicators of reef health. It engages in debris removal from the ocean and through ocean, and we do outplanting of corals and sponges to restore and rebuild damaged coral reefs. Right? So why should people care about the work that we're doing? Right? And one of the first questions that I tend to ask people is where does the majority of the Earth's oxygen come from? Right? The answer is the ocean. The ocean is responsible for more than 70% of all oxygen production on the planet. Most people think that oxygen comes from trees, and it does, but the greatest amount gets produced in the ocean. Right? The ocean is also responsible for cooling the planet, absorbing carbon dioxide from greenhouse gases in the atmosphere. I love the coral reefs. Right? Corals are, in fact, animals, and most people don't realize that. They're in the family Cnidaria, similar to the jellyfish. And you're going to hear more about jellyfish probably in the next presentation. Right? Coral reefs make up less than 1% of the entire ocean bottom globally. Right? That 1% protects over 90,000 miles of coastline around the globe. Right? It protects coastlines from the ravages of storm energy while also providing homes and shelters for over 7,000 species of fish and invertebrates. Now, hard corals, which are a primary focus, exist in a symbiotic relationship with an algae called zooxanthellae that lives within the coral. That algae is responsible for the coral having its color. Otherwise, their flesh is translucent and appears white because of the limestone skeleton that they also produce. When a coral is under stress, it will evict the algae and thereby exposing that white limestone skeleton. That is what is referred to as coral bleaching, because you're looking at the limestone skeleton. Right? Bleaching events are naturally occurring events, and they used to happen on 10 to 12-year cycles. And in that time frame, the corals would have an opportunity to recover from the bleaching event. But because of global warming, these bleaching events are occurring more and more frequently. And corals and coral reefs don't have a chance to catch up. Right? Coral reef organisms also exist in a nutrient deficient environment, which means that when a coral ecosystem is in balance, that very little waste is actually produced as a result of it being in balance and in harmony, because every organism eats and feeds off of every other organism. It is when that delicately balanced ecosystem is challenged that problems occur. And unfortunately, we're the problem. Right? We're dumping 7 million tons of plastics and debris into the ocean on an annual basis. Eutrophication, runoff, fertilizers, chemicals, pathogens, all poisoning our ocean. Right? But we can still fix that. Right? How we engage with the ocean matters. We get our oxygen and food from the ocean. Killing the coral reef means that it will die as a living organism and structurally collapse. And then the coral reef that protects 90,000 miles of coastline around the globe from 90% of all storm energy will cease to exist, and coastlines will be exposed to greater and greater amounts of storm energy.
Kramer, yay, the lightning bolt. You were almost done, I think. One of the things we were thinking about when we were putting this episode together was that coral reefs are ocean ancients because these animals take so long to form reefs and grow. Can you tell us a little bit about the time it takes, and which I think speaks also to why we need to protect them, because we can't just quickly wait for more to grow?
Well, coral reefs, like many of the other organisms that were discussed in previous presentations, are over 50 million years old, right? And it's difficult to say the time because each one of them grows differently. Right? So I'm passionate about corals, right? But in that, even corals, branching corals, boulder corals, plated corals, all of them grow at different periods in time. So the work that we do often is with branching corals because they grow quicker. You can take a coral fragment, maybe a couple of centimeters long, and put it up in the water column so that it can feed and have a full coral thicket within like a year's time.
Mm.
So by putting a host of them in the water, we can mass produce those corals in an attempt to restore them and get them back out on the reef as quickly as possible. But that's just branching corals. Boulder corals and plating corals take considerably longer time to grow. But we're working on techniques to make that happen even faster.
Wonderful. Well, I have so many burning questions about what those techniques are, so I hope we'll be able to turn back to that in the Q&A. So thank you so much, Kramer. We will definitely have you back here shortly for some more chat. And I hope that everyone is still putting lots of questions in the chat or using the QR code. And I'm very pleased to welcome our final speaker this evening to the Lightning Talk stage, Maria Pia Miglietta. Pia is an associate professor at Texas A&M University at Galveston, and her research focuses on the genetics, evolution, and systematics of a group of soft-bodied invertebrates called Cnidaria, the same name that Kramer just taught us, especially the immortal Medusa. And I have to say, immortality takes the Ocean Ancients theme to a whole other level. So I'm really excited to learn from you, Pia. Let's get your slides up and dive right in. Looks good.
All right, looks good. Let's go. All right, hi everybody. Today I'm going to introduce you to the immortal jellyfish, Turritopsis doorni, and I'll show you how it challenges most of what we know about death and life. And I'll change slide. Okay, so Turritopsis belongs to the phylum Cnidaria, and it's the less known cousin of corals and the big jellyfish that you see. It belongs to the class Hydrozoa, which is very understudied, and it has a very complex life cycle. So I'm gonna have a slide all for that. So normally, hydrozoans have a colonial form that you can see here, the colony, where the polyps reproduce asexually. So one polyp buds another polyp, and eventually at some point they will bud off the jellyfish, okay? And the jellyfish is the sexual stage of the cycle because it matures the gonads, usually around the mouth, and releases the gametes in the water, and fertilization is external. When the jellyfish releases the gametes, it gets old, aging, and dies. Now, Turritopsis dohrnii escapes this fate. So I'm going to add here those red arrows. And so what happens to the jellyfish of Turritopsis when they are under conditions, any stressful conditions, they don't die. So it can be temperature, or they are wounded, they're cut, the salinity is high, chemicals are in the water, they are starved, they don't die, and they revert back to the polyps, which are considered juveniles, right? And so for a jellyfish that doesn't die, then a new colony can be formed. And it does so going through an intermediate stage that's unique to the Turritopsis, that is called the cyst. This is how the colony looks like, this fuzzy stuff. This is on a root of a mangrove in the Caribbean. This is how you would see in the laboratory under a stereoscope. You can see the polyps, right? They have the stinging cells, just like any cnidarians. And if you can see on the left, they are budding off the little jellyfish. The little balls are the jellyfish. Those are the newborn jellyfish, the baby jellyfish, really tiny. We're talking millimeters. They have eyes and they're healthy. But if you starve them, this is how they look. Can you see how they are ugly, right? They're dying, they are shrinking, they're sinking at the bottom of the container, and it almost looks like they're decomposing. But if you wait a few hours, they become the cyst, this mysterious stage that is unique to Turritopsis. And the cyst, you can see, if you wait a few hours, this will last 1 or 2 days. It will sprout off the stolon, which is the anchor to the substrate. You can see all the cysts at the different stages right here. And then it will form a mouth and the tentacles, and it will become an actual polyp. That polyp will reproduce in many polyps and will form a huge colony. And when ready for that medusa that didn't die, that colony will form hundreds of new medusae that will be released in the environment. Okay, so this is a true metamorphosis, albeit in the opposite direction of the regular life cycle. Usually of the polyp that goes to forms the jellyfish, and then the jellyfish dies after reproduction. In this case, the jellyfish, when in unfavorable conditions, will go back to the polyp through the cyst. So Turritopsis effectively escapes death and aging. And senescence itself is a trigger for the reversal. In fact, if you keep the jellyfish in the lab and you let them grow old and they release the gametes and they're about to die, that aging process is a trigger for them to self-rejuvenate. So they don't even need an external stressor. They will do it by themselves. And the last thing that I'm going to say about these incredible jellyfish is that in the cyst, there is a process that occurs that's very important for humans, which is cellular reprogramming. That's when a cell is mature and becomes something else that is needed. So it kind of de-differentiates and becomes another mature cell. Now, cellular reprogramming and its genetic basis is very important for regeneration, for understanding stem cell behavior. And Turritopsis really represents this incredible system in very simple animals where we can study the genetics underlying these processes: aging, senescence, regeneration, and cellular programming. And you can check my website for more information or ask me questions.
Wonderful, Pia. So fascinating. The immortal Medusa. Can you tell us a little bit about how you got drawn into the world of the immortal Medusa, and what is it? Do you get to do a lot of work in the lab or in the field now? Kind of
Yeah,
how
so
did you get into this?
when I was an undergraduate student, I was in southern Italy where the species was found and was described. And we would play with immortal jellyfish. We would go diving and get the immortal jellyfish. And it was something that was normal to us. And then when I did my PhD, I started a small project on the evolution of this animal. And then nobody was studying this incredible animal. And I had questions. And so I continued studying from there. And we do field work in Panama mostly. So we go and we collect the animals one by one in the field. And then we have the laboratory component where we do genetic analysis of and experiments with the jellyfish.
Hello, it looks like our host is frozen for a short second. So at this moment, we're going to be bringing all of our guests back up on standby, and we're going to start our Q&A. Welcome, everybody. And I will keep an eye out for when our host returns. I know there have been some amazing questions that are popping up in the chat. So I'm going to take a quick peek through here. And before we even get into that, do y'all have any questions for each other? It was an amazing lightning talk.
Well, I never had an opportunity to hear from the other hosts. So listening to each one of their talks sent me to a number of different places. So trying to remember, right? And I thought the jellyfish talk was going to happen before mine. So I was waiting to hear about the jellyfish, but growing up in New Jersey and being able to see the crabs, the horseshoes, it's like learning more and more about them. Everybody's talk was just phenomenal. So not a question, just excitement to be in such esteemed company.
Yeah, that was so great. I learned so much and was so pumped up by all of your talks. I feel like, I'm going to be reading and delving more into all of this stuff.
I would love to kick this off with one of the questions we got from a 5-year-old viewer from, I'm going to, Sena, I believe. Senan, how many different kinds of ancient cephalopods were there, Dana?
There were thousands and thousands, more than 10,000 ammonites alone. Those are the ones with the coiled shells and also the weird shaped shells. And scientists just keep finding more of them.
That's so fascinating. Another amazing question for Rachel. So horseshoe crabs lay eggs on the beach. Oh, that is a word I don't know if I can pronounce. Are they? I apologize, you all. I am an event background, so science words sometimes are hard.
Chelicerates.
That word, yes. And do they stay in the ocean or do they return to the ocean?
Oh, that's a really good question. I'm really not sure. They are chelicerates. They're more closely related to spiders and scorpions than to crabs, even though they're called horseshoe crabs. But in terms of their evolutionary history, I really don't know if that was something that they returned to the ocean. But I don't know. It's a great question. I'll have to look into that myself.
That's awesome. Well, I have noticed it looks like Nora has been able to rejoin us on a new device. So I'm going to step out and let our lovely host continue with these amazing questions.
Hello, everyone. I'm so sorry. I disappeared exactly when I did not want to disappear, but I will try not to ask you questions that you've already answered. I would love to ask all of you whether your work on these ocean ancients has changed how you think about time in some way.
That's a great question. I just wanted to jump in, and I think that the thing that it changed for me when I started really delving into and doing research on the kind of deep time history of this group that I love, the cephalopods, is it really gave me a sense of time, just like the depth of time, like the sense of deep time, and that anything that feels like it takes a long time now, I have this part in the back of my mind that's like, well, it's much less time than it would take anything to fossilize.
That is true. Yes. Anyone else have any thoughts about time?
I think for me, it's just, sorry, go ahead. Just, the perception that there are so many different invertebrates that have a completely different life cycle than ours, and the way we perceive the world, it's so narrow compared to the variety of organisms out there and how they experience the world.
I think they make me think about time, but in a more recent sense, not looking back over the millions of years. But I am fascinated by the way the horseshoe crabs will come up just for a certain tide in the spring, and they all come up at the same time, and they come up at the high tide mark, they nest, and then they leave. So it could just be within a 20-minute or to an hour period that the beach is inundated with all of these crabs, and then they're all gone. And you would, if you weren't there and an hour later, you would never know that was what happened on the beach. And that synchronization around time and the lunar cycle is just fascinating to me.
And for me, it was really, happy moment to realize that we really found a very old kelp, and that more or less the timing of the DNA prediction said that the group should be there. But if you don't find fossils, you don't know for sure if those predictions are correct, the evidence for DNA. And then to find it and then realize that they must have been there even longer, then that's very exciting. And also living in California and seeing the kelps a lot, knowing that their record goes back so much further in time just makes me very happy.
Right now, for me, I'm at both ends of that spectrum, in that recognizing that coral reefs date back some 50 million years. In the grand scheme of things, when you look at things, it's like, well, how do you really quantify? You can, but how do you quantify as a human, as a person with a life cycle of a year, 100 years, 50 million years? But then on the other end of that spectrum, looking at climate change and what it is that we've been doing to the planet, and recognizing that we are running out of time. Right? It's a concern, and for the kelp, I know that we've been losing massive amounts of kelp on the West Coast as a result of proliferation of urchin.
Yeah, especially the purple sea urchin. If kelps are not doing well, they start to graze on the remaining kelps, and they end up making whole areas barren. And you then have zombie sea urchins walking on the seafloor looking for more food. And that's really, really bad.
Yeah. And on the Caribbean side, we've lost all of our urchins due to a pathogen that we allowed into the waterway. So we have a proliferation of algae, right? Because we don't have anything to feed off of it.
So all of these ocean ancients are so important in the ecosystems that they're in, as you've all shared. And we want to make sure that don't become things of the past if they're still with us today. What are some of the things that you recommend for people who want to make sure that these species thrive and continue and can be talked about in the future? Kramer, do you want to start?
Absolutely. Right. Events like this, right? The whole idea is educating everyone about all of the different things that are going on around them. And the more people are educated about what's going on, someone in the group that's in the chat is going to be excited by something that they heard from one of the presenters and say, I want to find out a little bit more about that, and or I want to study that so that I can make a difference. So things like this.
What about you, Dana? What do we need to do to protect cephalopods that are maybe not the 20-foot long one, but the ones that we have today?
It's an interesting group to be working on because they are, of the species that we have still in the ocean today, a lot of them are doing quite well compared to, other ecosystems and other species that are really struggling or really some even on the brink of extinction. Cephalopods are a really flexible, really adaptable group. And that's one of the things that has kept them around for 500 million years, is that they've been able to adapt. And so now we have all of these cephalopods with no shell, a very minimal shell in the ocean today, and very brief lifespans, most of them. And they make lots and lots of babies. And so actually when I was working on one squid species in particular, the Humboldt squid on the west coast of North America and South America, but it was on the west coast of North America that everyone was actually worried about an invasion, that it was becoming too abundant. And we were trying to figure out if that was true and how many there were. And we sorted out, I worked with a number of other scientists to sort out that it was really more of a natural range expansion. And there was no Humboldt squid invasion, as cool as the squid invasion would be. But it's an interesting group to be working in because they're, while there are individual species that are at risk, the nautilus, for example, which still has a shell, is on the endangered species list, needs to be protected from trade in its shells, which are often very highly desired. But in general, a lot of cephalopods are more like, they're kind of flexible. There's a lot that we don't know about them, and finding out more about them often gives us more information about how the ecosystem is doing. In some places, they might be becoming more abundant because their predators have been fished out of the ocean or are being caught in greater numbers. So they're in an interesting sort of in-between place, and it's a fun group of animals to study for that reason.
So interesting. And Rachel, what can you tell us about how horseshoe crab populations are doing right now and what threats there might be?
Actually they're monitored and managed by region. So where I am in the Southeast, I'm in South Carolina, the Southeast region of population is doing good. They've been documented as good. That was an assessment that came out last year. The Delaware Bay region is also good. Right now, the Northeast is neutral, doing okay. And the New York region was doing poor. And so it's something that it's important to, that's why it's important to monitor them all the time and get new data on them constantly so that they can update these assessments of the populations and see how they're doing. They're subject to habitat loss, which is one pressure that they face, and certainly climate change can play a role in that. And also it could be overfishing. They have to be monitored to ensure that they don't become overfished or are subject to overfishing and that our fisheries are sustainable. So, but in the Southeast right now, the population looks good.
That's wonderful. One of the questions from our audience, Rachel, was how do horseshoe crabs decide where to nest? So if there's habitat loss and they need to find a new spot, how would they go about hunting for a nesting location?
That's a good question. And there are lots of things that can factor into that, and a lot to do with the location itself, the slope of the beach, the type of sediment it is. And just recently in South Carolina, they found that they always thought crabs are just going to nest on sandy beaches that they can get access to. It's not blocked by some type of barrier. But now they're finding that they're nesting more in salt marsh and in muddier sediments than previously realized. So it's turning out it's more of a nesting habitat for the population, not just the sandy beaches. It's really, really interesting.
Oh, interesting. Yeah. I wonder if it's always been that way or if they've been adapting because they need to.
Good question.
Movie.
Mm-hmm.
Cindy, there are some questions for you about kelp. So there have not been a lot of kelp fossils found. So there's some curiosity in our audience about where one would be most likely to find a kelp fossil and why it is that they are so rare.
Yeah, that's a good question. And we're also wondering why that is. But where kelps grow are generally areas where there is a decent amount of oxygen in the water. If they die off and want to be fossilized, they have to end up in sediment that does not have a lot of oxygen in it. And that means that very likely, very often, if they die, they just rot away relatively fast because they live in oxygen-rich environments. So you need special conditions to get them preserved. But even in places where you have those conditions, they don't fossilize that well because they don't have a lot of compounds, although some of the parts are really stiff. They don't have a lot of compounds in them that help with preservation. And yeah, it is really a problem because as you saw in the diagram, there's so little fossil evidence that, yeah, we would definitely like to have more. And in the place where Jules Crania was found, people went back to that spot. And I think an older colleague of mine who went back to that spot said that it's now a parking lot for a fast food restaurant. So they're probably still there, but in the sediments, in the ground where we can't get to.
Okay.
Yeah.
So it was so lucky that one was found here in Washington, and we learned so much from just one fossil.
Yep, definitely. Very happy. And also Jim Goodett, the person who found, is an amateur paleontologist, and he looks around on the beach a lot for especially shelly fossils. So it was really good that he found these and thought, what could these be? Because otherwise, if it was anybody else, they would have just walked past them.
Well, I bet a lot of our audience looks pretty closely at things on the beach and in tide pools and where we go exploring, but that's definitely a lesson to be sure to do that because you never know what you might find.
Yeah.
Well, a question for you, Pia, from our audience is, if immortal jellies are immortal, how does that affect genetic diversity in the species?
Yes, they have also sexual reproduction. So in the wild, they can reproduce sexually, and that increases the diversity of the species. And then they can also do the trick of regenerating. So they can have both ways. So the genetic diversity isn't that affected because they can reproduce sexually.
Okay. And we were talking a moment ago, and I didn't give you a chance to answer, about how is the immortal jelly doing? Is it under threat? Is it doing okay? Do we know?
It's doing well, actually. Fun fact about the immortal jellyfish, it is introduced in pretty much around the world. In one of our studies, we showed that it was described from the Mediterranean Sea, and then now it's found in Panama, Pacific and Atlantic, in California, in the Gulf of Mexico, in Japan. So it's spreading. And we think it's traveling in ballast waters through boats. So it's human-mediated introduction. And we think that in fact, it was found in a boat that was traveling from one big port to another. And so we think that life cycle that allows the jellyfish to escape death if the conditions are not favorable may be a great advantage for them. And so they're traveling and spreading all over the world.
Okay. It'll be interesting to see what kind of changes that leads to in those ecosystems.
Mm-hmm.
Kramer, you've been a scuba diver for a long time, and I'm wondering if you can share what it was like the first time you dove and saw corals in the wild. That must have been something that inspired this work.
Ouch. I really would have preferred you to ask that question, or only because, like I said, I've been diving for 40 years, but when I started diving, I started as a wreck diver, right? And ignorant about the marine ecosystem for probably about 20 years, right, until I stumbled into working with an organization called the Coral Restoration Foundation. And then recognizing, one, that corals exist, right? That there are more than one or two, that there are hundreds of species, right? And then beginning to develop an appreciation for them and their importance. Right? They're beautiful to look at, right? But they do so many things, right, that people just aren't aware of, and in our ignorance, we end up destroying them. And they represent, like I said, less than 1% of the entire ocean floor is coral reef, right? So that's like the size of maybe the state of Colorado, right, for the entire planet, right? And we are killing it. Global warming is killing it. When we're trawling and overfishing, we're killing it. When we're on the beach, just stepping on and crushing these things that take 100 years or more to grow. It's I don't think there's a better way of identifying blissful ignorance. Right. In the beginning, not knowing, and then the more you learn, the more you're exposed to what the beauty is of that delicately balanced ecosystem and how everything exists in harmony with everything else until we get involved in the picture.
Well, thank you so much for sharing that, because you're right. They're beautiful, and sometimes that's what we see at first. But thank you for sharing all the super important things they do for the ecosystem, for people, for life on this planet. And there were a lot of comments in the chat from people who want to know how we can help. And you've mentioned some of those things. We need to have policy change to address some of those bigger issues with climate change and so on. We need to change our way of behaving and doing business. There was a specific question about those who dive and how they can be responsible divers.
Wonderful, right? Before I go into responsible diving, if everyone under the sound of my voice could just do one thing, which is stop using single-use plastics, right?
Great.
Wrapping up an apple inside of a container, then just throwing it away. 7 million tons of plastics we're discarding into the ocean every year. Being a responsible diver, the best thing that you could do is to learn how to be perfectly buoyant, right? So that you are floating neutrally in the water, not popping up to the surface, running into a boat prop, or crashing down onto the coral reef, destroying everything on the ocean floor, right? If you can find a way to establish peak performance buoyancy, that is the first primary thing that you can do to save our coral reef ecosystem.
Great. And thanks for providing also a really great ask for those who aren't divers or don't live near the ocean. Regardless of whether we live near the ocean, we can reduce those single-use plastics in all parts of our daily lives.
Can I point out something really cool? I love that neutral buoyancy, that call for neutral buoyancy. And it reminds me of cephalopods, because they actually achieved that perfect neutral buoyancy when they first evolved, like back before there were any fish or sharks swimming in the ocean. That's what they did with those shells, is they filled them with gas to achieve a like exact buoyancy. So I like to think of I'm also a diver, and I like to think of that diving perfect buoyancy as like, yes, I'm just like a cephalopod.
Amazing. Well, we're going to wrap up. I want to give each of you a chance to share one more thing. Maybe it's one more thing you want our audience to know about the animal or the topic that you shared, or maybe it's one more question that you're hoping to answer in your work as you learn more about these amazing species. So, or you can take it another direction. I'm going to start with Cindy.
Yeah, I said that nowadays, when you look at a holdfast of a kelp and you would open it up, if you're on the beach, you would see, got a little tiny animals crawling out of it or living in it, etc. And we actually, scanned the holdfast, the kelp holdfast, with very high Röntgen radiation, and made 3-dimensional movies. And when you go through those movies, you can see little tiny shells of little tiny animals that used to be living in those 32 million year old holdfast. And the work that we've done so far was mainly, hey, we found a very old kelp. I would like to go back to that mini world again together with my colleagues and write down who was living in those very old holdfast as well. But that's just something that I think is very cool.
That would be so cool. I'm going to ask Pia next.
I think I'm going to end with the questions that we have that we're trying to answer, and we are trying to understand how Turritopsis thorni evolved this capability. And it diverged from a very similar species that looks almost identical a few million years ago. And that species, if you stress it, it dies, while this species, if you stress it, it rejuvenates. And so understanding how these very similar species have those incredibly different capabilities is one of the main goals of the lab.
Kramer.
Okay. Well, Diving with a Purpose Collective Approach to Restoring Our Ecosystems is a nonprofit organization, right? So I would ask that anyone who is interested come and participate and learn about the organisms that exist in relationship to one another. And as a nonprofit, right, we're engaged in citizen science. Right? So most of these are PhDs here, right? Very intimidating. You're not intimidated by me, right? You get to come on a dive trip / excursion, learn about the coral reef, the ecosystem, and the organisms, and write the whole thing off as a tax-related expense.
Well, we will make sure that the website goes in the chat again, Kramer. That is a wonderful invitation, and I'm sure many of us are eager to do that. Rachel.
Boy, I don't know where to start. There are so many cool things about horseshoe crabs, but really there are cool things about so many animals out there. And really, I do love horseshoe crabs, but I love biodiversity. I mean, I'm partial to marine invertebrate biodiversity, but just get out there or go to an aquarium or zoo and take an appreciation for the biodiversity. Find something that fascinates you, and that I think is what helps us take care of the planet, is when not just what we learn something or we know a fact, but we are touched and we care about it. That's where we can really make a difference and work with others to make a difference and learn all you can, because there's so much more to the horseshoe crab story. There's so much more to my story, to all of your stories, and try to learn all you can about that story so that you're making informed decisions as you go along.
And Dana, you get to take us home.
You are all so inspiring. This has been incredible to be in this event with everybody. And I think that's my last thought, is that we are an ecosystem. We are part of nature. Humans evolved on this planet just like every other species. And I think sometimes, for better or worse, we tend to set ourselves apart a little bit and either feel guilty about it or feel superior. But we're part of nature, and we're made to make connections, just like every other species is in an ecosystem making connections. And I feel like that's something this evening has been all about. Like I can't wait to hear more about what was living in those holdfasts, for example, Cindy, because one of my first research projects was pulling apart holdfasts looking for tiny octopuses. And like, maybe there was a little octopus in them. That would be so cool. And I want to go diving with Kramer, and I just can't wait to hear what all of you are doing. And I think that perspective always gives me some buoyancy and some hope, remembering that we are all connected to each other and to the rest of the nature on the planet.
Well, thank you all so much. I also am so inspired by all of you and love all the ways you've piqued our curiosity about what more there is to learn about the animals in the ocean, and also pointed out all the important things we need to do to protect that life and conserve those resources and all that beauty and all the important things that they do for the ecosystem. And in the chat, I've seen so many comments from people who are learning new things and so excited about it. And there are kids in the audience who are really excited to be here. So thank you all so much. And I'm sure there'll be lots more kudos in the chat. So I want to thank you all and mention to everyone that if you didn't get enough of Lightning Talks, you can go to our website. We have all the past seasons and episodes there. And I'm also excited to announce our next episode, which is coming up on May 22nd. And the theme is spineless. So you can start to imagine what animals we might feature there. Certainly some of the ones we talked about today would also fit in. And if you've been, like me and all of our speakers, inspired by what you've learned today and the need to conserve our marine environment, we do hope you both look up all of the organizations and institutions that our speakers represent and consider a donation to the Seattle Aquarium to support our education and conservation research work. So thank you again, and we look forward to seeing you all again next time.
Thank you.